Good evening. Welcome to Within Sight. Welcome back if you've been here before. This is great. I think there's a lot of familiar faces in the audience. Thank you. It's nice to see you. Thank you for coming. Welcome if this is your first time here. I am Anna de Koning. I'm the Executive Director here at the Koch Institute. Last year we celebrated the 5th anniversary of our building where, as many of you already know, we combine scientists and engineers to develop new insights into cancer as well as new tools for detection, diagnosis, monitoring, treating, and preventing cancer. I'm excited to continue our celebrations by honoring some of our signature programs that we have here at the Koch Institute that accelerate new ideas and new technologies from our labs into the marketplace and into the clinic. So this evening we're highlighting our Frontier Research Program, which supports early-stage cancer research projects that very often do not qualify for traditional sources of funding, typically government funding. We do, of course, enjoy a good deal of government funding, both for established research projects as well as through our National Cancer Institute Cancer Center Support Grant. That they fund our Swanson Biotechnology Center, which which is the home of our excellent and essential core facilities here in the building. However, for these new, typical MIT, out-of-the-box, creative, groundbreaking type of ideas, we rely heavily on support from foundations, but particularly from philanthropists and individuals. And so we use gift funds to support high-risk, high-impact kind of projects. Some of these may flop, but we can learn a lot from our failures as well as our successes. But importantly, they all have real potential to change the world, or at least a field. Gift funds have also been key in building our community, because we prioritize projects that are both highly innovative, but also highly collaborative. And so people are encouraged to try something new with someone they have not worked with before. And that has been truly transformational. So all the projects are funded grassroots style by friends who contribute at every level, from modest annual contributions to gifts with a couple of extra zeros. Pooled together, though, this support adds up to a much larger impact. Now, I'm told we have given 76 Frontier Research Program Awards since we launched the program 10 years ago, and it's kind of brilliant that I am introducing this subject tonight when there happen to be 76 that have been awarded, when in a few weeks there will be more that get awarded, because we are in MIT Building 76, so it's just perfect timing. We've seen some great successes, and we'll highlight these as we continue our Within Sight series this coming fall. We will be featuring some projects that have moved out of the labs into the marketplace or the clinic. Tonight, we want to show you the impact of our Frontier Research Program, the impact it can have on cancer research, and importantly on the people, our trainees who conduct the research. So you'll hear from 3 presenters, as you probably saw on the poster, as well as just briefly, from 3 KI faculty members in whose labs they are doing or did the work. These 3 speakers were chosen because they really exemplify the spirit of the Frontier Program and actually the Koch Institute. And so I hope you will get a feel for the range of research that is enabled by these awards that is being done here at the Koch Institute. So without further ado, here is Frank Gertler, MIT Professor of Biology, to introduce our first speaker.
It's a great pleasure to introduce Madeleine Oudin. Madeleine began her scientific career as a neuroscientist. She got a degree in biochemistry from McGill, and then went on to King's College in London, where she got a master's degree in pharmacology and a PhD in neuroscience. I was lucky enough to recruit her to my lab. She joined the Koch Institute as a postdoc in my lab in 2012. And she's, in addition to being a great scientist, she's been very active in our community for our Committee for Community Life, which is an educational and public outreach program. And she's been very active in the community within the Koch and MIT-wide and in the scientific community in general. She's received a lot of awards, very prestigious awards, including a K99 Pathway to Independence grant. Sadly for me, she's just wrapping up her postdoc in my lab, and she's going to talk about the work that she did, so I'm going to leave that to her. But of course, I want to say that the Frontier Research Project program that supported Madeline and that she will be highlighting is particularly exciting to me because it employs engineering tools to further the work of a laboratory, mine, whose focus has been on more basic cell biology and basic cancer research. However, Madeline was not afraid to reach out to the engineers and find innovative ways to use those engineering tools and apply them to cancer-specific biological questions. Many of these engineering tools were not designed to address problems in basic biology, but instead have been repurposed by Madeline and others in the building to better understand the mechanisms that contribute to cancer biology, including tumor metastasis and drug resistance. Madeline?
Hi, everyone. Can you hear me? Yeah. In the back? Okay, great. Thanks. Thanks for the introduction, Frank, and thanks for being here tonight. I'm excited to share some of my work with you. So my work has been focused on breast cancer, a disease where tumors originate within the mammary tissues. About 1 in 8 women and 1 in 1,000 men will be diagnosed with breast cancer in this country in their lifetime. When the tumor is small, it is relatively easy to resect it and to find drugs to treat it. However, a lot of the mortality linked to breast cancer is when metastasis occurs. Metastasis is when tumor cells will spread throughout the body to other organs, such as the lungs, the liver, the bone, or the brain. About 30% of patients metastasize. There are about 150,000 people in the US living with metastatic breast cancer, and this results in about 40,000 deaths annually. So what does metastasis look like in a tissue? So here you have a schematic of normal ductal structure in normal breast human tissue. And here's actually a cross-section of human breast tissue. Here we have a purple stain for the nucleus of the cells, and the pink is the surrounding tissue and connective and the fat. As you can see, the normal breast is highly organized in these ductal structures. The tumors will initially grow inside the ducts, and will at some point break loose and take over the normal architecture of the tissue, as you can see here. The tumor cells will then disseminate through the vasculature to other parts, other organs within the body. So to prevent metastasis, we must better understand the process by which these tumor cells spread. So what actually attracts these tumor cells to leave their site, the primary site? So there's a lot of resident cells that can participate in this process, such as immune cells, which I won't mention today, but I want to focus today on the extracellular matrix. And the matrix are non-cell proteins that are surrounding the cells, both in normal and cancer tissues, that provide structure and support to these cells. So during tumor progression, there's a lot of changes in the components and amounts of the extracellular matrix. And so one of the most abundant matrix proteins in the breast is collagen. And you may know collagen because it's often seen on mammographies that are used to detect breast cancer. But I'm going to focus on today is fibronectin. So it's another ECM protein. As you can see from these images, fibronectin is largely absent in normal tissue. But in the breast tumor tissue, you have a lot more fibronectin, as shown by this green in the image. What's more interesting is that fibronectin actually accumulates around blood vessels. So you have high levels of fibronectin right around the blood vessels and lower levels further away. So you have endogenous gradients of fibronectin that are right next to the blood vessels. And so we wondered whether perhaps these gradients of fibronectin that lead up to the blood vessels might be acting as a gateway for the metastatic cells. So in our lab, we study metastasis in that it is driven by a protein called MENA-invasive, shown in this movie here in green. When cells have a lot of MENA-invasive, they move a lot more, they invade a lot more, and they metastasize more. Interestingly, we also found that patients with breast cancer that had high levels of MENA-invasive and high levels of fibronectin had worse outcome and more metastasis. So we asked whether fibronectin gradients could be maybe driving metastasis by attracting these MENA-invasive cancer cells into the vessels. So we've had this situation where we could see in humans there were gradients of fibronectin, and we needed to find a way to recapitulate this in a dish for us to be able to study it. We then also needed to find a way to study it in mice. Now, this was not easy, and a lot of the traditional biological approaches to these were really not great in order to really replicate these gradients that we wanted to study. And so this is where we had to turn to the engineering strategies that are being developed and repurpose some of the tools to apply to our biological question. So in the dish, we turn to microfluidic technology, which allows you to miniaturize devices to manipulate environments to study cells in. So we were working with a device whereby we could easily generate a gradient of fibronectin, just like the one we saw in the tumors. The cells, the tumor cells were plated in the middle channel. You can see we would do movies of the cells overnight, watch them move, and track their direction of movement using the forward migration index. If the forward migration index was positive, this meant the cells were attracted to the fibronectin. If it was negative, they were being repulsed. So what we found was that breast cancer cells that did not have any MENA-invasive were never attracted to fibronectin. Even at the higher doses, they were actually repulsed by it. But interestingly, the MENA-invasive cells were always attracted to fibronectin, even at the high doses. So after showing this in a dish, again, we always want to try and make sure we can validate our findings in a mouse. But again, how could I generate a gradient of fibronectin within a mouse? The microfluidic technology is not really applicable. So here, we collaborated with an engineer, postdoc Oliver Jonas, who's in Michael Sima and Bob Langer's lab. He had developed a device where there were reservoirs to load with drugs. The device would be injected in patients or in tumors, and the drugs would diffuse out over time in gradients. So this was perfect to apply this technology to our biological problem. So we loaded the devices with fibronectin that was labeled in red. We generated tumors, and we do so by injecting mice with human breast cancer cells in their mammary tissues. We wait for the tumors to grow. Once the tumors have reached a certain size, we expose the tumor, and we inject the device. The mice are anesthetized, and we can then perform imaging to look at how the cells are moving. And so this is what we get. So the device would be right here, and you can see a really nice gradient of fibronectin that formed on the edge of the tumor, shown here in green. And so what did we see? So we can see that in cells that are located close to the gradient, we can see these cells that are actively moving and spreading throughout the tumors, whereas if we look further away from the device where there's no gradient, the cells are not moving. So this was great to be able to use these technologies to show that gradients of fibronectin can attract tumor cells both in a dish and also in mice. So how do we actually treat metastatic breast cancer? It's good to understand what drives it, but it's also important to try and gear ourselves towards treatment. So chemotherapy remains the standard of care for metastatic breast cancer, with a wide range of drugs shown here in this list. The problem is that the response rate of the patients varies a lot, and even if the patients do respond, the time to progression can be very short. So the patients will initially respond but become resistant. So we asked whether perhaps the MENA-invasive cells, which drive metastasis, might affect the response to the drugs that are currently used to treat these metastatic tumors. So we started in a dish, and we looked at the survival of cells that had no MENA-invasive or the MENA-invasive. We treated them with a panel of the chemotherapy drugs that I showed you in the previous panel, and after 3 days of treatment, we looked at survival. Now, the MENA-invasive cells responded the same to most of the drugs except for one, which was Taxol, which is really kind of one of the main drugs used to treat metastatic disease. What you can see here is that at these doses of Taxol, which are similar to the ones that patients actually see in the tumors, the tumors actually see, in the patients, that the MENA-invasive cells had about twice as much of the cells surviving after treatment with Taxol at both of these doses. So again, after showing this in a dish, we wanted to move into the mice to see if this was also happening. So we generated tumors in our mice with the controls that don't express any MENA-invasive and the MENA-invasive tumors. We used a regimen of Taxol with 3 doses every 5 days, which is similar to what's done in the clinic, and we looked at a variety of tumor features. So first, we looked at the change in tumor volume. What you can see is that in the control tumors, there is indeed it slows down the growth of these tumors, so it's doing what we expect it to do. We looked at the cell migration of these tumors, and again, the Taxol did reduce cell migration, and it also reduced the number of metastases that can be found in the lungs. So again, the Taxol is working. However, in the MENA-invasive tumors, this was different. There's no effect on tumor growth. There was also no effect on the number of cells that were moving, and there was also no effect on metastasis. So how is this actually happening? Well, tumors normally grow by feature of uncontrolled cell division. So as one cell will divide into two cells. What Taxol does is it affects this cell division process. And instead of having two cells come out, the cells really struggle, and then one cell and the other one kind of dies off. And so over time, you're killing cells. The MENA-invasive tumor cells, shown in this movie here and this one, actually are happily able to divide in the context of Taxol treatment, and sometimes even divide into 3 cells. And so this indeed will then lead to continued tumor growth even in the context of Taxol treatment. So what I've shown you today is that the MENA-invasive cells are attracted to fibronectin, and this helps them metastasize. The MENA-invasive cells are also less responsive to Taxol chemotherapy. So what are we doing with this information? We are currently working with a company that's spun out of our lab called Metastat to develop commercial tests so that we can predict the likelihood of metastases within a patient and also help with drug treatment decision, if perhaps one patient might be more likely to respond to one chemotherapy versus another. So these are some of the things that we're doing to try and take our science more into the clinic. So with that, I just wanted to share what the Frontier and the Koch Institute have done for my career. So as Anna mentioned, the Koch Institute brings together biologists and engineers. This was a new experiment, and I'm here to say that it was a successful one. I came out, started here as a biologist, as a neuroscientist. I wanted to do something that had more of a clinical impact, and I wanted to work in cancer. I came to MIT, and I worked with a lot of different engineers and really opened my eyes to new ways of tackling cancer research. And I'm now happy to be putting all these things together into my own research, to be starting my own lab as an assistant professor at Tufts University in January. So with that, I'd like to thank my mentor, Frank Gertler, who's always supported me along the way. I've had great students that were supported by my Frontier grant who helped on these projects, great collaborators as well, without which I wouldn't have done this work. Our Biotechnology Center, which supports, again, all of our experiments, our funding from the Frontier program, as well as other sources. And thank you for your attention, and I'm happy to take any questions.
Any questions for Madeline?
Yes?
Oh, there's a microphone coming. If you could pass it down the road, that would be terrific.
First of all, congratulations for your success and good luck for your new adventure.
Thank you.
Angiogenesis has a big role in metastasis. Did you see any change on angiogenesis level between your normal cells and MENA-invasive?
Within the tumors themselves, not a huge, not a big change in the actual blood vessel density in the tumors. We have looked in breast patient tumors whether or not there's higher density of MENA-invasive near the blood vessels, and there is a small positive correlation. So, but that's not the only place that they go. They're definitely expressed heterogeneously in the tumor, not just near blood vessels. Yes.
Hold on, there's a microphone coming.
Is the MENA-invasive phenotype druggable on one hand?
Yeah.
And she's mentioned angiogenesis. I'm just wondering, is there any relevance to the immune profiling in that whole area?
Yeah.
Checkpoints.
So with regards to being druggable, I think that's definitely something we would like to look into to be able to drug it, for sure. So that's definitely an option. We've done a lot of work to try and characterize its function, where it's expressed, what turns it on as well, so we can better move towards that. So now we've really increased our understanding of the biology behind it, and I think we would like to move forward with that. With regards to the immunology part, it's not something we've explored yet, but I think a lot of the features or the characteristics of tumors that have high MENA-invasive, for example, this accumulation of the matrix, for example, has been shown to anti-correlate with immune infiltration, for example. So, I think there's definitely links with regards to the biology to that, but it's not something we've explored per se yet.
Thank you.
Okay, maybe one more last question. We have 2 more speakers. So.
Have you done any engineering of fibronectin to try to identify epitopes that are actually the attractive force?
Specifically with regards to the structure?
Well, just generating variants, mutations.
Yeah, so we've looked a little bit. So there's a part in fibronectin that activates specifically the receptor on the cell, the integrin, that will drive the signaling pathway. And in a 3D environment, that's actually not enough. So there's the signaling part, but there's also a structural aspect. So having just that little domain that signals is not enough to drive this process. So you need the signaling, but you also need the structure. So, and this really shows that the physical environment that these cells are in and how they take advantage of those different changes in cues is also very important. So it's not just a signaling aspect as well, which makes it even more interesting.
Okay, so I'm sure Madeline will be happy to answer more questions after in the dessert reception. I just wanted to mention that this was a really excellent project for us to highlight because the device that she mentioned from the SEMA and Langer labs was also funded by Frontier Funding, and it's actually already in the clinic now, so it's really a wonderful example of the impact we are having. Next up is Jackie Lees to introduce Monica Stanchu.
Thank you, Anna. I want to start off actually by thanking Anna and her team, not only for organizing this event and all the other events, but really for her stewardship of the Frontier program in general. We really couldn't do it without her liaisoning with both donors and also bringing projects together and support. So I really want to thank her. I also want to thank all of you for coming here tonight. We love your interest and enthusiasm and your support in general. Now I really want to bring the star of the show, which is Monica, and introduce her. So Monica recently graduated as a PhD student from my lab, and her work is focused on cancer biology, and particularly she's interested in bringing therapies to the treatment of brain cancer. Monica was originally from Romania. She conducted her undergraduate work at the University of Toronto, where, just like Madeline, she studied neuroscience and pharmacology. I think there has to be a magic formula there. And she's worked in several clinical research centers before starting at MIT as a graduate student. So Monica's, I think, is really taken remarkable advantage of MIT. She's a go-getter. She's been a remarkable scientist, but she also has a huge interest in biomedical research and healthcare entrepreneurship, and really thinking about how innovations profoundly impact healthcare discoveries and delivery and patient care. And while she was at MIT, in addition to her PhD, she took coursework at Sloan. She served on an executive committee for the MIT Hacking Medicine, and she helped, I think most importantly, organize a new course for my biomedical entrepreneurship that's really going to impact generations of scientists that come after her. So as far as the project goes, I think this is really perfectly fits the Frontier project formula. My lab had never worked on this. She came to me and said, I want to do a project on brain tumors. And I said, no, right? We don't work on brain cancer. We know nothing about it. We have no advantages. And she said, and I want to do an in vivo drug screen. And I said, boy, that's even harder. And it won't work. And are you crazy? And she kept coming back and back and back. And she more and more fleshed out this idea. And she found a collaborator in Mike Heeman's lab who had experts in screens. And eventually she broke me down, and I said, you can try a few preliminary experiments, and they got awesome data. And I said, great, let's write a Frontier Research Program grant, which got funded. And I think the project has been actually spectacularly successful. I have great hope that it's really going to actually have an impact on treatment with patients for brain cancer, and, not tomorrow, but I think coming down the road, and I'm going to let Monica tell you that story. So, Monica.
So, hi everyone, thank you for being here today. Today I'd like to tell you about a project that I was working on in collaboration with a couple of different labs while I was here, and I'd like to tell you about our efforts to identify novel therapeutic targets in malignant brain tumors, and specifically in glioblastoma. So first, to give you just a little bit of background to the motivation behind our project and why I think it's so exciting, I want to start off by talking a little bit about an area of very translational research that I'm sure you've probably heard a lot about, and that's personalized cancer therapy. So we know that not all patients respond the same to a particular therapy. And so the question becomes, how can we identify which patients will benefit from a particular therapy, and how can we treat an individual specific tumor? So our goal eventually is to be able to sequence a patient's tumor and identify particular mutations or vulnerabilities that it has, and then to target the therapy accordingly. Now, ideally, we would also want to have a biomarker or an indicator so that we could tell which patients are going to respond to that particular therapy and which are not. So, of course, we're now also beginning to understand that there are many factors beyond just the genetics, the genetic composition of tumor cells, and they can also influence tumor growth and response to therapy. But just to start off, and what we focused in on our project was to understand tumors at a gene level. So we can think of stratifying tumors in two ways. The first is to categorize them according to the area of the body from which they arise, their site of origin, because really tumors are a product of their environment. Oh, okay. So whether they arise from the liver, the lung, the brain, or the kidneys, or any other area of the body, we can use that to categorize tumors. Well, a second way that we're beginning to understand now is there are different features, and here I'm representing them by the different colors, that are shared between tumors that arise from different areas of the body. Because even though they may arise from different environments, they may use similar mechanisms to escape the normal checks and balances on cellular growth. And so it's becoming increasingly apparent that tumors that arise in different areas of the body can share these sorts of similar features, and these common features can be exploited and targeted for therapy across a variety of different tumors. So in the Lees lab, we are interested in understanding the mechanisms of tumor development and maintenance. And to do this, we use both in vitro and in vivo models, so cellular models as well as animal ones. And we combine genetic and pharmacological tools to really understand why tumors develop and how they're maintained. And this, in turn, can allow us to better target these tumors. So the project that I want to highlight today has been a collaborative effort from the beginning, as Jackie mentioned, and all the way to the end. And I've been working together with Christian Brown from the Heman Lab and Paul Boots from the Sharp Lab on this project. And we were interested in malignant brain tumors, specifically glioblastoma, or GBM. And this is the most common malignant primary brain tumor in adults. It's extremely aggressive and resistant to therapy, and the median survival is just a little bit over a year for patients with this tumor. So we were interested in identifying novel therapeutic targets for GBM, and even though over the last several years there's been a lot of knowledge about the types of biological background to GBM, there's still the most common course of treatment is surgical resection, followed by radiation therapy and then chemotherapy with an agent called temozolomide, or TMZ. And this survival curve shows the introduction of temozolomide, which helped to increase patient survival, but really these patients are only surviving less than 15 months. And so there's a great need to develop novel therapies for this kind of tumor. So our goal was to identify new genes that could be targeted for therapy, and one way to do this, you might think, would be to interrogate one gene at a time. And so to ask whether this gene, let's say gene X, is important for tumor growth, and to get rid of it, and then to look at what effect that has on the tumor biology. So in the simplest case, we can imagine that we start off with a mixed population of cells where we have some percentage that contain an inactivation of this gene X. And we can track this population of cells over time because we've also fluorescently labeled them. So here that's indicated by the green color. And over time, we can see how the proportion of those green cells changes depending on the genetic manipulation. So if that manipulation is beneficial for the growth of those cells, then the proportion of green cells will become enriched. If it's detrimental to the cells, then the proportion of green cells will be depleted, or it might have no effect at all, and then we won't see any change. So we can do this either in cultured cells, or we can genetically manipulate these cells in culture and then take them and implant them into mice and follow the growth of the tumors there. So to identify the pathways that are important in GBM and identify novel targets, instead of taking a one gene at a time method, we used an unbiased approach. So instead of asking just whether gene X affects tumor growth, we in fact took a pooled approach and put many genes together, and we let the tumor cells tell us which genes were the most important. And so here we start off with a pooled library, and we infect cells with this library such that each cell only contains one genetic manipulation, but because we're working with so many cells, there are several cells that contain the same genetic manipulation, and that's outlined here by the colors. And we follow this population over time, either in vitro or, as I mentioned previously, we can inject these cells into mice and allow the tumors to develop in the animals. And then we can see how the proportion of cells with each of these genetic manipulations changes over time, and we can quantify that, just like we would for the single gene case. So from this large-scale analysis, the gene that stood out was one called PRMT5. And as we can see from this graph, when we look at the level of confidence that we had across our 311 genes, PRMT5 came out as the top hit. Now, it's important to note that this gene is central to cellular activities, and in fact, without it, cells as well as entire organisms don't do very well. And so it's expressed at high levels in many different tumor types, which is proving to be very important. And this has led us, as well as others, to think that it has some important function for tumor growth. So after using this pooled screening approach and zeroing in on PRMT5, we set out to look at the importance of this gene for GBM growth and interrogating it more in depth. And so in our experiment, similar to the hypothetical example that I outlined previously, we can start off with a mixed population of cells and genetically manipulate a proportion of those cells in culture to inactivate PRMT5. And we can see that compared to the control seen here in black, each of the three different kinds of genetic manipulations that result in the inactivation of PRMT5 it results in a disadvantage to these cells. And so this is also true, actually, if we inhibit the function of PRMT5 using a small molecule inhibitor or a drug. Now, this is a different assay from the one that I showed you previously, but here we see that as that over time, even though the control cells that are not treated with drug are increasing in number, the cells that have been targeted with this particular drug are not. And so it was very fortunate, actually, for our studies that this new inhibitor had been developed just a couple of years ago, and we were able to use it for our studies both in vitro as well as in animals. So we can also test the effect of genetically inactivating this gene in our animal models, and we do this by starting off with genetically manipulating the cells in culture and then transplanting them into mice and then tracking the survival of these animals. So across the x-axis, we see here the number of days post-implantation, and on the y-axis, we have the percent survival of the animals from 100 to 0. And we see that compared to the control mice, which are implanted in cells with cells that contain no genetic manipulation, the mice that are implanted with cells that contain a genetic inactivation of PRMT5, indicated by the two red lines, which are just different manipulations, they in fact survive much longer. So we can also treat mice with this inhibitor that targets PRMT5, just as we did in culture. And if we treat them for a period of time outlined here by the gray bars, that indicates the dosing schedule, and then measure the volume of their tumors here on the y-axis, their tumors, the tumors that are treated here in blue with the drug actually grow to be much smaller than the tumors that are not treated with drug at all. And here I'm only showing a couple of the different animal models that we used, but of course we tested this in different models to verify our results. So what's interesting about PRMT5 is that it's actually expressed at much higher levels in glioblastomas, or GBMs, shown here, as well as in lower grade glioma tumors indicated by LGGs, as opposed to the normal brain. And this tells us that drugs that are targeting PRMT5 might be more efficacious in specifically treating the tumors without affecting the normal cells of the brain. In fact, PRMT5 is expressed at high levels in many different tumor types, and this is getting back to what I was saying before, that there are certain features that are common across different tumors that arise in different areas of the body. And so we believe that this uncovers a new vulnerability across many tumor types, not just in glioblastoma. Okay, so the next question that we wanted to ask was whether all cells would react the same way to this drug. So we tested a number of different cancer cell lines from different primary tumor types for their response to this drug. And the example graph that I'm showing here on the right shows the survival of the cells over many different doses of drug. And we can see that different cells have different sensitivities. So the cells that are outlined here in black are an example of a cell type that is much more sensitive, whereas the cells that are outlined here in the red dots are much more resistant to the drug. And what's interesting is that these divisions between sensitive and resistant did not track with the primary site from which the tumor is developed. So it wasn't as if all of the glioblastoma tumor cells were more sensitive than all of the breast cancer cells, for example, but there was a range of sensitivities regardless of where the primary tumor was. So this is really interesting because now we can start to ask whether we can predict which tumors are going to be more sensitive or more responsive to this drug. So remember, our goal eventually is to be able to sequence a patient's tumor and identify what mutations or vulnerabilities it has, and then target the therapy accordingly. And ideally, as I mentioned, we want to have a biomarker or a particular indicator that could help us identify a priori which patients are going to respond better to a particular treatment. So one might think that just the level of PRMT5 expression in the tumor would be a good enough indicator of whether that tumor is going to respond. But actually, it turns out that this is not the case, and it's just slightly more complicated. So we identified just such a biomarker that could help predict the response, and it could be potentially used as a companion diagnostic for drugs that are targeting PRMT5. So this biomarker is actually the ratio of expression of two genes, and both of these are binding partners of PRMT5. Each of these binding partners shuttles PRMT5 towards a particular pathway. One of these pathways, as it turns out, is very important for the cells to maintain their high growth rates. And so if the cells have high levels of binding partner 1, then they increase their flux through this pathway. And we've uncovered novel key details about the pathway and found that rapidly proliferating cells are actually addicted to this pathway. And when you take away their levels of PRMT5, they just crash. And so, moreover, we also believe that there are additional regulators in addition to just PRMT5. So this opens up many new avenues for therapeutic targeting in different tumors. So in this work, we've used both cellular models as well as animal models of glioblastoma, and we identified a novel pathway which enables tumors to fuel their growth. We defined a novel biomarker, which is particularly important because it can be used to stratify patients and therefore maximize the chances that a drug that's targeting PRMT5 could be effective at stopping tumor growth. And this has implications across tumor types beyond just targeting PRMT5 and also in GBM, which I think opens a very interesting area of research, and there's a lot to be explored there. And this project will be continued in different models of different tumor types in the Lee's lab as well as in the new labs of our collaborators. And so with that, I'd like to thank Jackie for her support and her commitment for this project from the beginning. As well as my collaborators and co-authors. This really was a completely joint effort, Christian and Paul. I also want to thank my thesis committee while I was a graduate student, Mike Heeman and Tyler Jacks. All of the work that we did was with great support from the Swanson Biotechnology Center. And we're really very grateful for the support that we received from the Frontier Awards because, as Jackie mentioned, this was an early stage, very risky project, but it had the potential for high impact given the dire need for development of new therapeutic strategies in glioblastoma. And it's really allowed for a fruitful and what we hope to be a long-standing collaboration between our different labs, and it highlights the interdisciplinary environment here at the Koch Institute. Thank you, and I'd love to take any questions you might have.
Questions for Monica?
Hi, is the inhibitor that you used in the clinic today?
So it actually, a form of it, a slightly modified version, is in phase 1 clinical trials currently, not just for glioblastoma, but a number of different solid tumors. So it's a dose escalation trial, yeah. We're watching that very closely. That's with GSK.
Does GSK have a good biomarker to stratify their patients?
They don't yet.
That's exciting. Any other questions? Hold on, can you wait for the microphone? Sorry.
So at the beginning, you nicely show how the tumor have similar factors and then different.
Yeah.
The biomarker that you identify that couples with yours is the same for each tumor or is different?
So we think that that biomarker could be used to stratify many different tumor types and determine whether those tumors are going to be sensitive or resistant.
So both biomarker is common on these tumors.
So what we're calling a biomarker is actually a ratio of the expression of those two genes. So it's not that there are two separate ones, but only when you combine their expression levels do they turn out to be predictive of the response to therapy.
Did I miss or you didn't say which one is it?
I didn't say very specifically.
Might not be protected.
Quite right.
Hot of the press research here tonight. One quick final question, or shall we move on to the final speaker? It might be good to move on, actually, in the light of time. Okay, so finally we have Michael Sima. Yes, thank you, Monica. Michael Sima, Professor, David H. Koch Professor in Engineering.
That's right.
After 2 professors of biology, is going to introduce our final speaker.
So I'm here to, whoops, can you hear me? Yes, I'm here to introduce Katerina Mansa-Vanu. Huh? Don't try it, it's Katerina. She's originally from Athens, Greece, BS in Engineering Sciences from Harvard, which we don't count against her at all. She entered MIT as an HST student 3 years ago. We were just talking about it today, 3 years ago. She's passionate about access in healthcare, and you're going to hear about a little bit about that tonight. She was awarded a prestigious fellowship last year, the Tata Fellowship, with a number of other students here at MIT, but it's quite a very special group of young scientists. Her research concerns ovarian cancer and, in particular, access to therapy there, and you're going to hear a little bit about that. I have a few words to say about the Frontier program, and I have them to thank for this, or this program to thank for my introduction to ovarian cancer research, and specifically Tyler Jacks, who introduced me to a number of very important ovarian cancer researchers, Mike Beerer at MGH and Marcelo Del Carmen, who continue to be collaborators with me today and have helped in Katerina's research. You might be surprised it's hard to fund translational research, and you don't do that with necessarily with NIH funds, and even that's getting more difficult. So these types of funds are really important to us here. This has been a my research is all in translation. I try to do basic science in the clinic as best I can. A lot of that starts with preclinical research, and that's what the Frontier program is about. Little did I know when I started this, which was this type of research in ovarian cancer, that my family would be faced with trying to survive ovarian cancer. And just a few years later, and I credit us doing that was how much I've learned about ovarian cancer in this research. I had no idea what my research, how it would prepare me for that experience and prepare my family. I'm happy to say we're still surviving ovarian cancer, and the prognosis is really great in our case. The differentiating factor in our situation was luck and access, and so that's what Katarina is going to tell you about tonight. Katarina?
Thank you, Michael. Checking that you can hear me. Thanks so much for that intro, Michael. Like Michael mentioned, I joined his lab because he's famous for working on really translational work and building products that actually affect patients' lives. So we represent the engineers in this building. We are found on the 6th floor, and we basically build a lot of technologies, working with the biologists and all the other basic scientists at the Koch, trying to bring all the new science that is done in this building over to the clinic and to the people that suffer from various diseases and cancer. And so today I'm going to be talking to you about my project focusing on building origami implants to beat metastasized cancer. And like Michael said, we are looking at a specific kind of metastasis that occurs in many malignancies at an advanced stage, and it's metastasis that spreads throughout the abdomen, known as peritoneal metastasis. So this diagram here shows you a picture of what the patient looks like when they have this kind of tumor spread. What happens is that basically their entire abdominal organ space is seeded with tumors of various sizes, and they also get filled with this fluid shown in red, this malignant fluid that contains floating cancer cells everywhere. And this is them at presentation. This kind of metastatic spread affects many people in the US, more than 50,000 patients in the US. And specifically, as Michael highlighted, it's a very common spread for ovarian cancer patients. More than 75% of women facing ovarian cancer are diagnosed at this stage, known as stage 3 or 4. And as Michael also hinted at, survival statistics really vary depending on when you pick up the disease. So if you pick it up early, you face really good prognosis, but if you pick it up at this stage, the 5-year survival is very low. Now, a lot of work in this building is focused on early stage diagnostics, and we're hoping that in the future we're going to be able to catch this disease as early as possible. But right now, many patients with many malignancies face this clinical picture. So what our work is focused on is on helping them deal with this disease. Now, how do these patients get treated in the clinic? First, they have to undergo surgery to remove as much of this tumor as possible from their body. And in that process, they may also lose parts of their healthy tissues that may be coated with tumor and are not necessary for survival. For women, that means removing the ovaries and their uterus, but also maybe parts of the intestines. It's a very radical surgery. And it's very hard to do a really good job and remove all of this tumor from this space. So you can imagine you actually get left behind with microscopic nodules, either because they're invisible or because it would be compromising the patient's safety to remove them, or because it's just circulating free tumor cells that are still in that space. So these patients have to get chemotherapy after the surgery. And they can get the chemotherapy through their systemic circulation, through their veins, intravenously, or they can get it locally. And this local chemotherapy is known as intraperitoneal or IP chemotherapy. The way it's done is that the patients get implanted with a tube, a catheter, into their abdomen, in their belly, which is left in place for multiple weeks, during which they receive multiple infusions of chemotherapy. So they go back to the hospital every few weeks, and they get pumped with 2 liters of fluid, shown here in blue, that contains basically the maximum amount of chemotherapy that they will survive through for that infusion. And then they have to be given 3 weeks of a break to basically bounce back and be healthy enough again to be able to undergo this again. And this happens for 6 cycles. That's the recommended dosage. You can imagine that this is a very challenging technique. However, data has shown, both in clinical trials, but maybe more importantly in the clinic, which is what this data is from, that there is a marked survival benefit for patients with ovarian cancer stage 3 who undergo any sort of IP, local chemotherapy delivery after their surgery. You can see here in blue that these patients fare better in their overall survival than patients that only get systemic or IV therapy. At the same time, in that same paper, the authors analyzed how many of those eligible patients will actually get any sort of IP chemotherapy in their regimen. Now, this data is coming from 6 national cancer centers in the US. So we're talking the top hospitals, and they are ready to go resource-wise and training-wise to do this and support the patients that go through this regimen. You can imagine that that's far from what you would get in an average hospital if you needed to get treatment anywhere else. And with that in mind, you can see that really never in this time duration did IP chemotherapy get given to more than 50% of eligible patients in these centers. And in fact, adoption varies dramatically across centers. So some hospitals really do IP chemo, and some hospitals will give it to fewer than 5% of eligible patients. And it kind of is, like a draw of whether the physician you're being treated by believes in IP chemo or not, which is a problem. But why is the adoption so bad? So I showed you a little bit about how this is done. You can imagine it's logistically very complicated and requires really highly trained individuals to do this, so nurses who can support these patients. But on top of that, and perhaps more dramatically, it's just a very toxic regimen. So data has shown that more than 50% of patients, so the majority of patients, can't pull through these full 6 cycles of IP chemotherapy. They drop out and they have to be switched to systemic chemotherapy. And the main reasons have been identified to have to do with the way with which the chemotherapy is given, so the catheter and the port they have to get in their body, and also the fact that they get these high drug doses, the maximum they can survive through, and it's just too toxic. And so our work is focused on basically replacing this kind of very dramatic regimen with an implant, a device that is inspired from a lot of work that has happened in the CIMA lab over the years, and also inspired from the ability to create origami-inspired deployable structures, and can be placed in the body to deliver continuous low-dose chemotherapy, be placed with minimally invasive surgery, match the mechanical properties of the tissues around it so that you minimize how much you irritate the organs around it, and you minimize side effects for the patient, and is safe for the patient to have in their body throughout this course of treatment. So how would we put this device in the body, and why is it called an origami device? Well, that was one of the key aspects of the design that we had to deal with. How do you fit this kind of implant through tools that the doctors use today, which have diameters of the order of magnitude of millimeters? So you have to be able to thread something through that and still get a surface area that's big enough to cover this entire space and all of the microscopic tumors you may have left there. And that's where origami came in the picture. And so we started playing with different ways to create geometries that can result in big structures, think like big, large sheets, but can collapse into these tiny structures that you can safely pass through these tools. And then ideally at the end of treatment, be able to remove them. And so this is the inspiration for the kind of the big picture image of what these devices would look like. Now, a very important question to ask is, okay, which patients would benefit from this work? So we already know from clinical data that the patients with peritoneal metastasis, and specifically ovarian cancer patients with peritoneal metastasis who get IP chemo, the ones that do the best are the ones that had the best outcome in surgery. What that means is that the surgeon was able to remove all the visible tumor, and all that was left behind was basically non-visible tumors. And so if you think about it in terms of scale, we're very interested in this region where tumors are sub-millimeter in size, not visible to the naked eye. And so in our lab, we did work basically trying to recapitulate these sizes in vitro and seeing whether we could implement a continuous chemotherapy regimen and still be able to benefit those patients. Remember, we are reducing the dose at any moment here. So instead of going through these really high infusions, we want to replace them with a lower continuous dose that's always present. And so a graduate student that's now graduated from our lab, Laura, did a lot of establishing all of this work, and I joined her project near the completion, and what we worked on was creating these ovarian cancer cell aggregates, which we will call spheroids because they look like spheres, and you can see them right here. And we focused on two sizes, one that resulted in 100 micron sized little nodules, and one that resulted in 200 micron sized nodules. What that means is 0.1 millimeter and 0.2 millimeter. So both of those are basically invisible to us. And the difference between them is only 0.1 millimeter. And what we found that if we treat them in vitro with various dosing regimens that basically recapitulate this high dose kind of periodic treatment that we see in the clinic, or replace it with continuous low dosing, we get very interesting results. So for the 0.1 millimeter sized spheroids, there was no statistical significance in the outcome of these different regimens in terms of doing better than each other in treating. So we were able to treat effectively with all of these regimens. But when we hit 0.2 millimeters in diameter, just 100 microns more, we started seeing differences across these dosing regimens. What we saw and what we learned was that A, you can't really replace these periodic high-dose infusions with the same total amount of drug given over a longer period of time at a lower dose and necessarily expect that the larger tumors will shrink as well. But we found that we can actually give more drug in total, but still keep the dose at any moment at a lower value and do better than these periodic infusions, which was very promising because that's exactly what we're hoping to be able to do with the kind of implant that we're developing. And so we set off to also start building these structures, and we developed biomaterials that match the mechanical properties or are close to the mechanical properties of the tissues around them, and that will hopefully be well tolerated in vivo, and that allow us to tune how we deliver these chemotherapeutic drugs from them over time. And you can see we can actually, by changing parts of the composition of the biomaterials, we can actually get various release rates of drug over time. But is it actually possible to build something that will fit in the abdomen of the patient and deliver so much drug over time? Well, this is kind of the size of the samples that went into that diagram. And so I've done a little bit of math and looked at the surface area of release through which drug was delivered. And what this translates to, if we want to essentially replace the current total amount of drug given to the patient over the same duration of time, is imagine a very, very thin sheet of 14 by 14 centimeters in size, which is actually not a bad dimension to place in your body. So this is very promising. We can actually do this. Now, I showed you some numbers in the beginning about the clinical need for this work, and we talked about the picture in the US, but I wanted to take a step back and also look at what the need is in less resource-rich settings. So there's more than 2 million patients every year in the developing world who face this kind of peritoneal metastatic spread. And the Frontier Award allowed all of this work in the past year, but it also allowed me and gave me a chance to go ahead and explore how we could make a difference in the life of those patients as well. And so, as Michael mentioned, I am now a Tata Fellow. There's a center here at MIT, the Tata Center, that focuses on challenges in India and finding solutions for them. And I have been able to establish collaborations with top national cancer centers in India and work there to understand how the doctors there would view a solution like this for their patients. So what does that mean, and how has the Frontier changed the course of our project? As Michael also said, I'm part of the HST program, and the HST program is a collaboration between the Harvard Medical School and MIT. And what that means is that they basically take engineers and train them with some clinical training to be able to innovate healthcare. So we take a full year of medical school classes and we spend time in the clinic like medical students. We scrub into the OR, we are present with appointments with doctors and physicians and their patients, and we learn how to take history and write notes. We're part of the healthcare delivery process. So we really get a very good picture of what the challenges are in the clinical setting. And that ties in very well with all the translational work that Michael does. And so I've spent months of my PhD scrubbed into the OR and really seeing these surgeries and the patients and what it means to have this kind of cancer and what it means to remove all the cancer from the body. And I've seen the patients receive their chemotherapy and deal with their side effects. And I've seen that both in Boston at Mass General, but I've also seen it in the middle of India in national cancer centers there. And I've been truly inspired and in awe trying to get a sense
sense of how much our work actually could hold promise for all of these patients in reality. And that would never have been possible without the funding that we have received from the Koch Institute and specifically the Frontier program. So with that, I would like to acknowledge everyone that has helped me in this journey so far. And I'd also like to thank my funding sources that include the Frontier for making this work possible, and take any questions you may have. So.
Oh, here's someone. Sarah, microphone. Oh, is there a microphone?
Right here.
Does this process lend itself for other forms of cancer, like prostate or bone cancer or other, or is it just limited to the experimental work you're doing now?
So that's a great question. The kind of cancer spread that I'm talking about, you actually see in cancers from the GI tract. So small and large intestine, gastric cancer, stomach cancer, sometimes lung cancer can cause this metastasis. Definitely ovarian cancer. You can see it with all cancers of the gynecologic tract. I don't think prostate cancer has a high incidence of peritoneal metastasis, but the implications for all of these diseases and being able to treat in that space are huge for cancer. And if we talk a little bit beyond cancer, it can actually be a very big deal for other diseases that could benefit from delivering drug in that space. But yeah, there's a lot of implications for a lot of other cancers. Yeah, absolutely.
Actually, I think Michael has a startup that is now in clinical trials for bladder cancer, right?
Yeah, that's right.
Yeah. Another question over there.
Thank you for that presentation. It takes me back to my early days in New York at Sloan Kettering with Maury Markman and Charlie Levenbach, who were the ones who were struggling with these peritoneal catheters. They all got scarred up. It was such a hard and miserable experience.
Yeah.
It's nice to see a next evolution of thought. Makes me wonder why we didn't think of that 20 years ago ourselves. But I'm wondering, so you could do this in advanced disease where the peritoneum is kind of spread because there's fluid in it. Mm-hmm. But were you doing this in an adjuvant setting where the surfaces, the visceral and peritoneum are stuck together, how do you expect to infiltrate that thing in there? And aren't you going to run into the same issues with scarification? And then the other thing is how much of that drug is going to reach how far?
Yeah. Those are actually both excellent questions. So the questions are, A, if you don't have all that fluid in there, how are you going to get the implant in there, and how are you going to, and the second is, how are you going to get all the drug to go throughout all that space? And those are actually excellent questions in the design process. So the answer is that we are working to build this so that it can address those problems. In terms of not having as much fluid in that space, there's as I'm sure you know better than I do, there's a small volume of fluid there at any time. And in fact, the circulation of that fluid is in part a reason why the metastasis spreads so much throughout the cavity. So we are actually now doing tests in mice and healthy mice, trying to see whether we can, with a little implant like that, get drug everywhere. So we're in the process of answering it. Now, if we see that it's not possible to have a single device and do that, we are exploring options such as having multiple smaller things that are placed in key locations, let's say near the diaphragm where there's a lot of metastasis, or near the liver, and treat those. But basically, it's a work in progress of addressing those, but they're definitely at the core of the design process for this work. So
I suspect it would be the first time you use it where you're going to get the best distribution. Mm-hmm. Because every time you put something in there, it's going to scar off that neighborhood. So you're going to lose quadrants very quickly.
Yeah.
Unless you can get it in on the first try when you still don't have all that fibrosis.
Yeah, and hopefully we can minimize the fibrosis, both with the material we use, possibly adding things to the surface that are antifibrotic, and making sure that it mechanically matches the tissue as much as possible. But absolutely, very good points, for sure. Thank you.
Okay, I think there was a question in front of you. Did you want to ask a question? Or no? Behind you? Okay.
There's one back there.
Oh, over there.
My question is not that much about the technology, but you mentioned that even the hospitals that are equipped to treat effectively nowadays don't do so.
Yeah.
Is it well understood why doctors choose not to go with the treatment that has been proven to be better? And what makes you, why, I'm guessing we should be hopeful that your new technology will make people more willing to use it. Why is that?
That's also a great question. So actually the authors of that study pointed out a lot and were shocked to find the differences in adoption across centers. They themselves didn't have a good answer as to why one National Cancer Center gives it to 4% of the patients and another one gives it to 70% of eligible patients. And they actually found no differences in terms of adoption when it comes to things such as race or socioeconomic background, which could play into whether the patient chooses to have a more drastic procedure like this or not. So really what it seems to boil down to is physician preferences and whether they think that the toxicity and kind of all of the side effects and the complications associated with this are worth the survival benefit. Now, in clinical trials, there's a lot of work being done trying to understand how you can actually reduce these toxicities, how you can find the best dose for IP chemo. Is the survival benefit really applicable to all the patient populations, or which population is the best one to give this to? And so it's a very complicated picture. I mean, it ends up being very much a call of the doctor whether they think this patient should get it or not. And I think like you point out, if the technology made the treatment less kind of drastic, less toxic, less complicated, then I don't see why the adoption kind of obstacle wouldn't be reduced that way. So I really think that we are suffering here from a very bad technology for something that kind of scientifically and on a scientific basis has a lot of promise for having results and already yields results despite how bad the technology already is. So, yeah.
All right. Katerina didn't mention it, but her paper, one of her latest papers was just published on Monday, and MIT did a nice write-up about it. So if you're interested in hearing more, you can go check it out. Let's thank Katerina and actually all of our three speakers. I just want to close by saying that we are really proud of our trainees, their passion, their dedication, their commitment to doing good in this world. As I think we clearly illustrated this evening, our trainees come from many different places and backgrounds, and the KI is a place where we cherish and celebrate that diversity, and our trainees' successes are also one of the many ways in which we measure impact. So please join us again this fall when we continue our With Insight series. We will highlight some more frontier research projects that actually made it into the marketplace, and also feature our Bridge project signature funding program that is basically a collaboration between the scientists and the engineers here at the Koch Institute at MIT and the clinicians and researchers at the Dana-Farber Harvard Cancer Center across the bridge, hence the Bridge project. And actually also supported Katerina's and Michael's project. The Bridge Project program will be on October 5th. We don't have the date for the other one yet, but save the date for October 5th. And now it's time for dessert. I'm sure the speakers would be delighted to answer more of your questions. And thank you for coming, and good night.